For patients diagnosed with advanced refractory cancer, standard treatments frequently fail. Surgical resection, ionizing radiation, and conventional chemotherapy often yield diminishing returns against highly mutated tumors that develop sophisticated resistance mechanisms. When these broad clinical guidelines are exhausted, families often look to clinical trials. However, trials enforce rigid exclusionary criteria. If a patient has complex comorbidities or an advanced disease state, they are disqualified, leaving them with the impression that they are entirely out of options. There is a viable alternative. Through the FDA's expanded access pathway, patients can pursue a single-patient investigational new drug application, or IND. This legal mechanism allows an individual to obtain a custom-manufactured, AI-engineered personalized cancer vaccine. This pathway has strict prerequisites. The patient must face a serious or immediately life-threatening condition, and all standard National Comprehensive Cancer Network guidelines must be exhausted. Furthermore, the patient or their advocate must be prepared to step into a demanding project management role. Securing a custom biologic is an exceptionally complex, multidisciplinary endeavor. It requires mastering and executing a rigid operational protocol to translate abstract machine learning concepts and stringent legal frameworks into a clinical reality. Step 1 is mapping the patient's unique genetic architecture. The development of a targeted therapy relies entirely on securing an accurate biological baseline to feed into the artificial intelligence pipeline. Standard pathological analysis is insufficient here. The advocate must instruct their clinical team to order comprehensive genomic profiling, utilizing advanced next-generation sequencing platforms. There is a critical trap at this stage regarding tissue preservation. Routine diagnostic biopsies are preserved in formalin-fixed paraffin-embedded blocks. Formalin chemically cross-links and degrades nucleic acids, destroying the exact high-fidelity DNA and RNA required by the machine learning models. The advocate must explicitly mandate that the harvested living tumor cells be flash-frozen or transported in specialized proprietary media. Concurrently, a peripheral blood draw must be taken to serve as a healthy genomic reference. Without preserving uncorrupted biological data at the very moment of biopsy, the entire downstream computational process is structurally compromised before it even starts. With the physical tissue secured, the workflow moves from the laboratory to the clinic. The patient cannot apply to the FDA directly. The absolute prerequisite for this pathway is a licensed physician willing to serve as the sponsor investigator. Securing this partnership is difficult. Oncologists in high-volume clinics frequently decline due to the overwhelming administrative burden and legal liabilities involved. When pitching the oncologist, the advocate must avoid vague requests for an AI vaccine or machine learning treatment. Such terminology lacks regulatory context and will likely be dismissed as unscientific speculation. Instead, use this specific, structured framing. Standard NCCN guidelines are exhausted. We want to partner with you to pursue an individualized therapeutic cancer vaccine under a single-patient IND via expanded access. Immediately mitigate their concerns about paperwork by confirming that you have engaged a specialized third-party navigation service or compassionate use consortium to handle the FDA application and manufacturer logistics. Acquiring a bespoke therapy requires strategic coalition building. The advocate must translate a desperate medical situation into a highly structured regulatory request. Once the clinical team is assembled, the genomic data generated from the biopsy is sent to a precision laboratory and fed into a proprietary machine learning pipeline. A single tumor may harbor thousands of mutations, but only a fraction will provoke an immune response. The AI algorithms simulate the physical binding affinity of these mutations to the patient's cells, identifying the precise neoantigens most likely to trigger a targeted, lethal T-cell attack. Translating this digital blueprint into a physical vaccine exposes a severe systemic hurdle. Because these custom neoantigen vaccines are investigational, health insurance companies universally deny coverage. This chart breaks down the comprehensive financial liability. Between the high-throughput sequencing, the AI computational processing, and the clinical administration, the total cost typically ranges from $100,000 to $119,000. This funding must usually be placed in escrow before manufacturing even begins. This timeline visualizes the unavoidable temporal burden. It takes roughly 4 weeks to process the sequencing, followed by 4 to 6 weeks for the computational design and AI predictive modeling. The finalized blueprint then enters good manufacturing practice laboratories for custom synthesis. This physical construction and rigorous quality control testing requires an additional 12 to 14 weeks. In total, families pursuing this route must secure massive liquid capital and endure a grueling 4 to 5 month wait from biopsy to the first dose, all while the underlying cancer continues to progress. While the physical vaccine is being manufactured, the clinical coalition must execute the required regulatory bureaucracy to legally administer the biologic. First, the physician must secure a letter of authorization, or LOA, from the manufacturer. This document grants the FDA permission to reference the manufacturer's highly confidential master files to assess the underlying safety of their proprietary technology. This checklist outlines the critical components of FDA Form 3926. The physician must systematically prove a dire prognosis, the total exhaustion of alternative therapies, the biological plausibility of the AI platform, and detail a rigorous clinical monitoring protocol. Concurrently with the FDA's 30-day review period, the protocol must be cleared by the hospital's institutional review board. The patient must sign an informed consent document that explicitly outlines the risk of life-threatening autoimmune toxicity. This dossier is a meticulously structured legal argument. It is designed to prove that the existential threat posed by the refractory cancer justifies the unknown risks of an unapproved bespoke biologic. Following the extensive preparation timeline, the customized vaccine is shipped under stringent cold chain logistics from the manufacturing facility to the treating hospital for administration. At this stage, a critical biological complication arises. The vaccine acts as the ignition, training CD8+ T cells to hunt specific neoantigens. However, the tumor microenvironment deploys immunosuppressive proteins that act as brakes, deactivating the attacking T cells upon proximity. To overcome this defense, the expanded access protocol frequently mandates synergistic therapy. The experimental vaccine is co-administered with an FDA-approved checkpoint inhibitor. Working in tandem, the checkpoint inhibitor neutralizes the tumor's suppressive signals, allowing the AI-guided T cells to successfully infiltrate and dismantle the malignant mass. Expectations must remain grounded. While proprietary models can now rank tumor mutations with a mathematical precision that was previously impossible, early phase 1 data primarily demonstrates safety and immune activation. The therapy remains a sophisticated experimental gamble, with no guarantee of broad survival benefit. By refusing to accept the limits of standard clinical guidelines, patients and advocates can navigate severe systemic bottlenecks. Executing this meticulous workflow remains the only viable mechanism for accessing the most advanced tier of precision oncological care outside of a clinical trial.